Okay, people have about 30 seconds. People coming in, please come in. People are staying outside. We get going in a minute. I'm Martin Friedlander. I'm at Scripps Research Institute most of the time, and I have the pleasure of organizing this session this morning. So I'd like to thank the organizers, which I guess would include myself, for inviting me to organize this. And I'm especially grateful to the speakers. We've sort of limited the number of speakers because we'd like to have more discussion than talking. Unfortunately, Kristen Baldwin, actually fortunately I think, because I'm told we have a better speaker now than Kristen. Okay, so Kristen is stuck on the East Coast, and so we have Valentina Lasorda, who's a senior postdoc in her laboratory, who will be speaking to us about IPS potency and trying to grab younger cells from younger people rather than older people. And then Sean Buchanan is here as a staff scientist in Lee Rubin's lab at Harvard. He's going to be talking to us about how to revitalize the cerebral vasculature in your brain. Since the two other speakers' combined age together is probably still less than mine, I was actually parabiosed before the talk today, and so I will have the benefit of receiving some of the factors you'll be hearing about from Sean. The purpose, we initially thought of organizing this session about vasculature, instead of replacing damaged or diseased vasculature, with the idea of working up and enhancing central nervous system components, which include neurons, but of course also glial cells. And it's sort of evolved into something a little more interesting, I think, not necessarily from my perspective, but from your perspective. And that is, we're really interested in emphasizing the role in a microenvironment and the state of the cells that you put into diseased tissues. And primarily in this case, we're focusing on the central nervous system. And also to emphasize that the vasculature is not simply a conduit through which red blood cells and platelets and serum trickle, but rather is composed of a variety of cell types, many of which can assume the role of stem cell-like cells, or probably more like progenitor cells, since they're a little more differentiated. So I'd like to sort of emphasize again that the stem cell biology isn't necessarily simply stem cell or cellular replacement. It's not necessarily cell replacement therapy, but rather probably is a very significant paracrine function that these cells produce. And I think you can hear that from each of the speakers. So I'm going to stop talking and ask Valentina to come up and speak. As I mentioned, she's a senior postdoc in Kristen's laboratory. She trained with last year's keynote speaker, actually, Elena, I'll mispronounce this. Catania, okay. And she's going to be talking about examining the effect of aging on IPS potency, and also to use stem cells to study what many of us are utterly exasperated by. That is, we can sequence all this information, genetic information, identify variants, but then how do you translate this into something which you can actually use to develop therapies for? So her last 3 manuscripts have been in Nature journals. Do you only publish in Nature journals? No, okay. And it's going to speak to us today about reprogramming approaches to studying aging and genetic risk for vascular disease.